You're listening to Beyond the Endpoint, a podcast from the Duke Clinical Research Institute. In this series, we're demystifying clinical research and answering your pressing questions about health and healthcare. You can find our full episodes at dcri.org/BTE.
Welcome to Beyond the Endpoint. I'm Emily O'Brien, and I'm joined by my co-host, Manesh Patel, who, like me, is a faculty member at the Duke Clinical Research Institute. We're coming to you from the 2025 American Heart Association Scientific Sessions, one of the most exciting gatherings of the year for the field of cardiovascular research. This annual conference brings together global leaders from across the scientific community who are driving innovation in cardiovascular health and research.
Thanks, Emily. That's right. We're here at the AHA Scientific Sessions in New Orleans and carved out a few moments to just check in with each other and our Beyond the Endpoint listeners. There's a lot of energy this year at AHA. There's new data, some thought-provoking discussions happening in every room, and just all around getting to see people that you've interacted with in the past, so it's pretty exciting.
Yeah, so speaking of exciting things, we have some news to share. You are the new president-elect of the American Heart Association. So you will serve as the association's volunteer president beginning in July of next year, July 2026, once the current president's term is complete. So congrats on what's an amazing honor, and I'm sure will be a really exciting component of your career.
It's humbling. It's an honor that you get to do because others have given you opportunity, and with that opportunity, hopefully there's a responsibility to make a difference. I often say when I get to do this conversation about being president-elect, I'm hoping that we can live up to our impact goal and our mission as we talk about everything from those discoveries to how we get them to our patients and our communities. So today's special episode, we're switching things up in honor of the AHA. Instead of just discussing recent headlines, we're sharing highlights from the sessions. And there'll be sessions we've attended. There's going to be some stuff that caught our attention. There'll be some surprises, but I actually think there's going to be things that a lot of our audience may be reading in the news or reading online. And so we want to get into some of that and delve into some of the data. So, I thought maybe I'd start by just saying the first session, the opening session, was pretty exciting. There was an intro from Joe Chikwi, who's the session's chair. It's going to set the stage of all the different ways we're going to interact at the American Heart Association. But then it broke right into the first late-breaking clinical trial that had a lot of interesting sort of science around the evolving world of cardiometabolic health. New drugs for lipids, new drugs for fatty liver and obesity. Any things there that caught your eye?
Yeah, well, one of the things that I love so much about this meeting is that it is very diverse in terms of attendees. People come certainly from all over the world, but also from all different backgrounds and different focus areas in terms of their research. I spend all my time in population health phase 4 post-marketing stuff, and I get to come and be essentially a student for phase 1 findings, phase 2 findings, things I would never get to really interact with in my professional life. And so one of them I thought was just incredibly cool, CTX310, the gene editing therapy that targets ANGPTL3. And the cool thing about this one, I thought, is it's delivered as a single IV infusion, and amazing effects in terms of LDL lowering, triglyceride lowering, and at least in this preliminary, very small cohort of 10 people, no adverse events or any safety concerns. Certainly a lot more work to do, but the idea of having a one-time infusion to help manage triglycerides and cholesterol, I thought was super cool. So I hope we'll eventually get to some work if it moves to phase 4.
Yeah, that's right. Talk about the thing I just said about making sure these discoveries get to people where they are. That story is really interesting to me in that we're getting more and more personalized, but we're using genomics in a way I didn't think we would. And, I was maybe skeptical about. So PTL3 was actually identified as a potential therapeutic opportunity and a gene editing opportunity from a community. I forget exactly where the community is. I think it's between Rome and Tuscany, but there's a community in Italy that has people that live extremely long lives without having a lot of atherosclerosis. And so doing gene studies in those individuals, just like the PCSK9 story, found that these individuals have really lower LDLs and not as much atherosclerosis. So the target was genetically identified through something we observe in the population. That's interesting. And then the CRISPR-Cas9 story, which I think is going to evolve at this meeting and every meeting, because gene editing has such huge opportunity and such complexity to figure out if it's safe and can you do it, right? And so for gene editing, what they showed us were 12 patients that got it. One had some liver enzyme elevations, and we'll have to follow that because these editing agents, we always worry a little bit about that. And then the real opportunity with this target, as you said, is it's everything from FH to high triglycerides and everything in between. And so not only is it a target that might affect people that have LDL issues, but triglyceride issues and a variety of others. So as just a big opportunity that's quite exciting. We'll have to see if it can come to fruition. In the same session, I was excited about, there were other agents like that, a triple agonist for GLP-1, a GIP, and then something was a fibroblast growth factor in the liver to really reduce fatty liver. We'll have to see how those things play out, but we saw a couple of things there. And then a big outcome study called Vesalius, which looked at PCSK9 injections and showed in primary prevention and in high-risk primary prevention, reduced not only cardiovascular events, not only MI, stroke, but also CV death and all-cause death. I think the question I posed on one of the media outlets was like, is there an LDL number too low? And then it's just a question of how do you get there, hopefully with lifestyle, not always with pills and shots, but
We've been asking that question for a while.
Yeah.
One day we'll answer it.
I've been starting to tell patients, like, just keep trying to get it as low as you can because there's no downside that we can see of.
Well, that trial was very close to my heart. We did an FDA-funded ancillary study looking at fitness for use of EHR data to ascertain outcomes. That one I thought was a really nice opportunity to work with a sponsor that had appreciation for the meta science. And like, if we were to do this trial and rely on existing data, like what would we find? I think more sponsors should do that if we really want to push forward some of these more pragmatic approaches.
Oh yeah, I think I'm very interested in this too because I spent some of my time doing clinical care and some of my time doing research. I don't know why they have to be so separated. I wish they could work together with the same data, same action, maybe even some of the same education. And I think actually as we get to more and more tools with people like you or data scientists, population health, help us understand that, but actually access to the data, having it in a way that's usable for the clinician and the patient when we're seeing each other. I often say that my hope for AI, as we have already had AI conversations on this podcast, is that we'll have more human to human time and less human to computer time. And I think patients and clinicians would like that. Well, let me get to another set of studies that I was kind of interested in, get your take. And you hear about blood clotting studies at the HA, and you might say, well, I'm going to skip that one. That's not the most interesting. And I get it. I understand why. The ones that I saw in the late breaking session 2, which I thought were of interest, were around atrial fibrillation. Just reminding our audience, that's the most common dysrhythmia of the heart, where the top part of the heart is irregular, the bottom part's still squeezing, but that irregularity lets a clot form. And patients who have that clot form then are at risk for stroke. So historically, we used to use warfarin, and then about 15 years ago, we had some new drugs we call direct-acting oral anticoagulants. And now we've had strategies to ablate that. Electrophysiology colleagues can go in there and stop the AFib. Or they put a device in there called a left atrial appendage occlusion device where some of these clots live in the heart and see if they can do it for people that are at high bleeding risk. And we saw 2 trial results that will affect our clinical practice, I thought. And the first one, maybe the most interesting and one that we didn't think we saw coming, was a head-to-head trial out of Germany in about 1,000 patients where the device was put in high-risk patients, CHA2DS2-VASc, that's a risk score for stroke of 5, a bleeding score of 3. So high-risk patients, average age 79, a lot of comorbidities. They put the device in and watched them without being on anticoagulants. They might have been on an aspirin, and compared them to the anticoagulants. And the primary endpoint was supposed to be stroke, systemic embolism, cardiovascular events, and then bleeding. And even with that combined endpoint, which we usually don't do, the device didn't win. The blood thinner won, even though those patients didn't want it. So at least makes us take pause in those older patients that are high risk. And then the other study was ablation. After ablation, people often ask, can I come off my blood thinner? And the guidelines to date have sort of said you have to be on it forever because of your risk. But if you stop the AFib with ablation, does that change? And investigators in a multinational study out of Canada published in the New England Journal found that, in fact, when you're lower risk, CHA2DS2-VASc 1 or 2, the stroke risk was very low. And so for the take home for the audience, I say you need 2 things to be on a blood thinner for AFib. First, you need AFib. So if we get rid of AFib and you don't have it for 16 months, maybe you don't need to. And then you need risk. You have to have enough comorbidities to risk-benefit analysis. And I'll just say, this is an area where we did all of these trials and it took 10 years. But back to your first point, we've been using these devices and these blood thinners in practice for 5 to 8 years. So better data science opportunities might teach us some of these signals. I still believe many times they'll have to be randomized. We can get into more heated discussion about that. But at least in this regard, I think this was interesting. Any thoughts on that?
Yeah, I mean, I worked for a number of years with John Puccini on Orbit, and that was a very active time. All these new DOACs, and we're trying to see why they're used, why they're stopped, and
what's the practice, what's the life?
Yeah, and also when someone has something like nuisance bleeding, and you see this reaction. One of the more interesting pieces to me was a sub-study that we did that looked at patient understanding of the condition, and what we found is that for many patients, it hadn't been really explained to them in a way that apparently made sense because they didn't quite grasped that stroke was even the major risk of atrial fibrillation, which may have impacted some of their preferences in terms of treatment. And so every time that we do one of these preference studies, I'm always amazed at how, first of all, maybe patients have different perspectives and experiences than we think, but also that the provider risk avoidance philosophy, it can differ quite a bit from what patients want, especially, in these complex populations where stroke and bleeding risk are both high.
Yeah.
That's the piece that I think we need much more attention to.
Yeah, no, I totally agree. And I think it's actually a place where we excel in some of the research you have done and others have done, where we are actually trying to get to the person that matters the most, which is the patient or the person who has atrial fibrillation. We're taught do no harm. And so there's a lot of bleeding avoidance. There might be stroke avoidance, but then there's some patients who say, well, I don't want to go in the hospital, have a GI bleed and get 2 units of blood. I think that looks like a stroke. So, I think this is really an area where, just like better therapeutics for blood pressure and cholesterol, there's a need for better antithrombotics in this space. And in that space, we actually on Saturday saw Regeneron actually has a monoclonal antibody that they presented in patients that is kind of interesting, as it tells us about how patients might respond to another sort of target factor 11 in some of those patients. So that was interesting. And I guess that's the stuff that I think a lot about. But if you said to me out of AHA, what's going to get a lot of interest? And maybe we should talk about it now, because I know it's going to cause some antibody reactions with you anyway. But I'm going to say there's a trial called the DECAF trial. Now, before I tell you anything about the DECAF trial, tell me, this is an interesting exercise, what would be your worries about anybody who tells you about some relationship between caffeine and heart rhythm problems?
Life is about balance. We try to talk about shared decision making, try to make a risk benefit kind of determination for a lot of our choices. And the risk of cutting out caffeine, I think, would just be too high to me. The benefit is so much higher than any downside. So it's one of those that I'm not wanting to read too much about. And I'm interested, because I know you wouldn't have brought it up if there weren't something there, but I'm a little bit nervous to hear the results.
Yeah, listen, I don't want to stop caffeine. Are you kidding? Obviously, people know me. I drink a fair bit. And the reason I brought it up, because I know you're so passionate about this, is historically, these kinds of studies have been observational and they've been associative, not causal. And so when we say in the nurses' health study, we saw that nurses that drank caffeine live longer. Well, it may be because they were able to get caffeine and others can't. So it's always complicated to solve, right? What's really interesting and why I think it's making it to the late-breaking stage is because the DECAF trial randomly assigned 2 things. So what happened is patients came in who had an atrial arrhythmia and they were getting cardioverted out of it, back into normal rhythm, which is not uncommon in clinical practice. Somebody goes into this rhythm and then they say, what can I do to stay out of it? And we're struck with this conversation of, should you be telling this person, should you drink caffeine or not? What we've seen from the trial design is several hundred patients who had this cardioversion were randomly assigned to a recommendation of liberal, drink as you were, versus really, really watch it and trying to do things to watch those patients, and then follow their atrial arrhythmias. And there was no difference in atrial arrhythmias, if not a little bit numerically less with the caffeine. And when you look through the observational data and some other smaller randomized trials, you start to see a signal that certainly doesn't look like it's as bad for atrial arrhythmias as we might think.
Oh, well, this is a reason to celebrate. Right? We should have started with this one. I would have been in a great mood the whole time.
So I would say the real interesting thing about DECAF, and they named it well, is that the study does 2 things. It says, we're going to try to answer it with randomization, which I like in this scenario when you can control it. Obviously, you need many bigger numbers to see, but atrial arrhythmias come about a lot. You can monitor heart rhythms a lot, so you get a lot of data points, and at least they don't see a signal. That doesn't mean that we know for sure, but it certainly doesn't have a very clear harmful arrhythmia effect on atrial arrhythmias. Obviously, I'm not speaking about others, and we need to be very cautious about overinterpreting. But at least I would say, coming out of this, I think we all have a sigh of relief to be able to say we're going to potentially be able to do some of the other caffeine issues.
There were celebrations with that trial team when they saw those data. And they were excited.
I think actually the trial team had seen the prior observational data, then seen a lack. And I think they realized hey, if we do a randomized study and demonstrate that, then that'll actually be a pretty powerful one and probably break the internet. So, those are good things to have. I will highlight at the AHA, it's always great to think about our young stars in our space. And so a couple of people in our organization are presenting results of interest we wanted to highlight. And there's a young faculty member, I'll say junior to mid-career faculty member named Marat Foudim, who's presenting the first in human gene therapy trial for patients with heart failure and preserved ejection fraction. That's where your heart's squeezing normally, but you still have heart failure because it's stiff and you get a fluid backup. And this trial called MUSIC-HFpEF looks at a modulation of CA2A, which is a calcium channel in the heart muscle, and it looks at the intramyocyte calcium trafficking in the heart. And so early phase, first in human, but great to see that experience and starting to say, can we do gene therapy for heart failure and stuff? And then Neha Pagadapadia, who we've talked about, she's presenting a sub-study, or at least a subgroup of the KARDIA-3 trial, which is a zilebesiran, which is a small interfering RNA molecule that you can be testing against standard of care hypertension to see if it improves outcomes, is going to be studied. And they looked at it just to see blood pressure in patients with and without kidney disease. All, again, quite interesting issues that I think people will certainly find interesting.
Since we're on the topic of young or, mid-career folks who are sort of getting started and like making a name for themselves within the community, we actually had an instructor yesterday who gave a really fantastic presentation at our Q-Core Day at session. You may know him. His name's Rob Califf. He's instructor of medicine.
Yeah. That speaks to our awesome appointment and promotions committees.
That's right. Well, I said to the audience as I was introducing him, he has about 1,200 publications. So we'll see how APT looks at his CV. And then he said that the reason he has yet to be promoted is that he needs to submit an individual development plan, and he just hadn't gotten around to doing it. So we'll see. We'll watch his trajectory closely. But he had some incredible comments, and I thought just a really thoughtful perspective, which he said himself, he's not a government employee anymore, not looking to be promoted, not really looking for a job. So he's speaking openly. And we were talking about the implications of HR1, the One Big Beautiful Bill for cardiovascular care, and some pretty sobering statistics that were shared, particularly with respect to its impact on rural populations, which I know is an area of really interest and passion for many members of the Q-Core community. But he ended with such a sort of powerful call to action for scientists to begin to advocate more for policies that promote cardiovascular health in their communities. And we're seeing so many changes. And I know we love to focus on the science and don't love the idea of getting wrapped up in politics, but it was just really powerful to hear someone with that level of perspective reflect on, his career and to say, hey, it's important for all of us to stand up for what's right too. We're a community that has a lot of expertise and people care about what we think. And it's important to share that knowledge, both one-on-one with friends and family and our community members, but also to the extent that we can through, various mechanisms like social media. And that's one way that we can ensure that our voice is part of the conversation.
Yeah, no, I think it's a great conversation to have. And I think it's an important one because fundamentally, no matter what side of the aisle you're on or how you think about this, our communities in rural America have to be healthier. And if we're not putting policies in to make not just rural America, all of America, including sometimes our cities, healthier. And the policies that lead to food security, the policies that lead to opportunity to get access to healthcare, I think everyone's on board with those ideas. The question is how we're going to do it.
Yep.
And so I think that's a real vigorous opportunity for us to say, this is how we want to do it, or this is what we're doing, or we're testing, or we're willing to advocate based on what we're seeing in our own practice in our communities. And sometimes we don't always have the opportunity to recognize that we can have an impact. So it's really powerful to hear Rob say that and encourage people to do that. And I think sometimes these meetings, whether it's with him or an opening session or a scientific or a junior faculty presenting something, it's invigorating at these scientific sessions for me because it brings us back to what I'll say often at these things, which is, life's about service and purpose, and the opportunity to see yourself make that difference is pretty fun. And so it's everything from the puppy booth where you get to feel your blood pressure, see if that changes, to science on hypertension, to advocacy, to what's the possibility of a gene therapy that might change all of the cholesterol and how would we test it. So that's pretty exciting. We're excited to be here. Thanks for doing this podcast with him.
I was a double visitor to the puppies.
I should tell you
5 minutes wasn't enough.
So I was saying to these guys that in 2019, I was the session's co-chair, and we were trying to figure out how can we make the meeting more exciting. And there was some, although not great, observational data that petting puppies lowers blood pressure. We wanted to increase awareness. And at that time in 2019 in Philadelphia, we asked the local shelter, and they brought these puppies and said they want to get them all adopted. So we opened up the puppy booth. It had the longest line. Even I said the snappy one got taken the last day, so they all got adopted. And then the next day we had large randomized trials on sodium, changing the sodium content. So the opportunity to go from something that feels fun and relatable to what's the next wave is pretty exciting.
So that brings us to the end of this special episode of Beyond the Endpoint, coming to you from the 2025 AHA Scientific Sessions. These conferences represent the cutting edge of clinical research, and we're happy to have brought you into a few of the conversations that grabbed our attention.
Yeah, really fun having this conversation with you, Emily. And thank you all for joining us. As always, we encourage you to share your thoughts or recommendations for things we should be talking about by emailing us at beyondtheendpoint@duke.edu.
And don't forget to subscribe to the podcast on SoundCloud, Spotify, Amazon Music, or Apple Podcasts. You can also connect with the Duke Clinical Research Institute on LinkedIn or by following at DCRI News on X and Blue Sky. We hope to see you next time on Beyond the Endpoint.
This has been Beyond the Endpoint. Resources related to this episode are available on dcri.org/BTE. If you have any burning questions about clinical research or healthcare, drop us a line at beyondtheendpoint@duke.edu. We'd love to address your questions in a future episode.